Hello dear learners, in our today's video we have to understand the concept of DNA replication. Before we start on with the DNA replication, we should understand some of the basic terms related to replication. What is the meaning of replication? We know that according to central dogma, if the DNA strand is again formed from the parental DNA strand, this process is called as replication. If this DNA converts into RNA, then this is the process called as transcription, and if this RNA converts into the protein, then it is referred to as the translation. So the process of formation of new DNA strand from the parental DNA is called as DNA replication. According to the experiments which were done by Messelson and Stahl, we come to know that DNA replicates semi-conservatively. The term semi-conservatively refers to that the two DNA strands which are together, suppose if I make these strands like this, then the two DNA strands will first separate, and after that the new strand will be formed from each of these strands. Means here each of these strands will be acting as the parental strand, and from those parental strands the new strands will be formed. So here the conservation of parental strand in the progeny is only half. Means half of that is the parent strand and half of that is the newly synthesized strand. So we say that there is a semi conservation of the parental strand. However, there were the alternative theories also which were proposed for the DNA replication, one of them being the conservative mode of replication. Conservative mode of replication says that the complete parental DNA strand will remain intact and it will then generate the new strands which will be then double stranded. Means in the new progenies the parental strand would not go. The parental strand would remain intact, but when the new progenies will be formed, in those progenies the parental strand would not be available. There both of these strands in the new progeny will be newly synthesized. Then there was another theory also which was put forward, and that is dispersive mode of replication. In the dispersive mode of replication, it was said that the original parental strand got divided into many pieces, and then each of them, either through semi-conservation or through conservation, was developing the new progeny. Means the whole of the parental strand got dispersed and then it started forming the new progenies. So there are three modes of replication, but out of them only one mode of replication is accepted and is true, that is semi-conservative mode of replication, which was put forward and explained by Messelson and Stahl's experiment. Now let us talk about the two types of replication which we have to study in our syllabus. One of the replication is replication in prokaryotes, the another being the replication in eukaryotes. So first of all, before starting with the replication in prokaryotes, let us try to compare the replication of prokaryotes with that of eukaryotes. The first important point is that the prokaryotes have a closed DNA, means their DNA is not open at any of the single points. But in eukaryotes, the DNA is open. In prokaryotes, the DNA is circular as well as double-stranded, like this one. So here we have two strands and they are in circular fashion which are not open from anywhere. But in eukaryotes, there is linear and then there is double-stranded DNA like this. The prokaryote's DNA is located in the nucleus-like region wherein the replication will take place. Means there is no true nucleus, but there is a nucleus-like region inside the prokaryotic cell, which is also referred to as nucleoid. In the eukaryotes, there is a specific, well-defined and membrane-bound nucleus inside which the DNA is present, which is in linear and open form, and double-stranded form, of whose the replication will take place inside the nucleus itself. The DNA of the prokaryotes while doing replication is kind of monorepliconic. The term monorepliconic refers to a single ORI site in the DNA through which the whole of the replication will take place. Means only one of the ORI site is involved in the replication. So this whole unit will be replicating together, and this whole unit will act as a replicon. So that's why we say that the mode of replication in prokaryotes is monorepliconic. But when we talk about eukaryotes, the mode of replication in eukaryotes is polyrepliconic. It simply means that there are many ORI sites and there are many replicons. We can say that because there are many replicons and because there are many ORI sites, so the replication will not take place in the continuous fashion. Because there are many ORI sites, so repeated replications at new places of the DNA will take place. Please remember that these replicons inside the eukaryotes are referred to as ARS, which stands for autonomously replicating sequences. So that's why the eukaryotic DNA is polyrepliconic, but the prokaryotic DNA is monorepliconic. Now let us come to the details of the replication in the prokaryotes. Let's move on to the replication of prokaryotes now. As I have already told you that the prokaryotic DNA is double-stranded DNA, but it is closed and it is in the circular fashion. It only has one ORI site, which is located over here. Now let us talk about this specific ORI site. The ORI site consists of approximately 250 base pairs. There are 3 to 4 repeats of GATC sequence in this ORI site. Suppose once GATC sequence here, then here, then here, then here. So there are 3 to 4 repeats of GATC sequence. The initiation of replication in the prokaryotes will begin when this GATC sequence is methylated, and that too very specifically when this A region or when this adenine of this GATC sequence gets methylated. When all of the GATC sequences of the ORI site will be methylated, that time we will say that the ORI region is fully methylated, and that is a clear-cut signal for the initiation of replication. After the methylation has taken place, now the replication will begin. For the beginning of replication, a protein which is referred to as DNA-A protein or DNA-A ATP will come and bind to the ORI site. The main function of DNA-A protein will be to melt this ORI region and to uncoil this ORI region so that it forms a shape of bubble. So it will be forming a shape of bubble so that it opens. It has to open because the new enzymes which will come will be recreated in this space. So after the action of DNA-A protein or the DNA-A ATP, the melting of the ORI site has done, and now first enzyme will be coming and binding to this part which will start the replication. The name of the enzyme is DNA-B helicase. Now the DNA-B helicase is a very special protein because it is a homohexameric protein. As soon as it is recruited on the ORI site, it will recruit the main DNA polymerase, that is DNA polymerase III. The working and the action of DNA polymerase III, that is the polymerization activity of DNA polymerase III, will be from 5' to 3'. This DNA polymerase III also has the exonuclease activity, wherein it can cut the wrongly incorporated nucleotide, and that is it has the direction of 3' to 5', means it can do the proofreading from 3' to 5' later on. So first it has to do the polymerization activity from 5' to 3', and later on it can do the proofreading activity from 3' to 5'. Now let us look into the details of the structure of DNA polymerase III. A typical DNA polymerase 3 is made up of approximately 9 to 10 subunits. Out of those subunits, let us first see the beta clamp subunits. There are two beta clamp subunits which I have made here in pink color, 1 and 2. As the name suggests, this will hold on or clamp the parental strand of the DNA so that the holding of that parental strand is done and the replication can be taking place smoothly. Then there are 5 to 6 gamma complexes which are attached at the base of the beta clamp. The main function of these gamma complexes is to open and close the two clamps so that the strand can be held on or left. Then above them is the main enzyme, that is core DNA polymerase. It is only one in number. So this core DNA polymerase is that enzyme which is actually going to do the replication. The holding of the strand will be done by the beta clamp, and the opening and the closing of these beta clamp will be done by the gamma complexes. Now there is one another unit which is called as tau unit is present on the top, and it acts as a dimerizing unit. When the two strands are to be replicated during the same time, so one of the DNA polymerase here and the other DNA polymerase will be working here, and the two will be dimerized together by the tau. So this tau unit will be having a very specific function which we will learn when we start learning the process of how the replication will take place. In DNA polymerase 1, the gamma complex is absent. So if the gamma complex is absent, so there is no flexibility. The opening and closing of beta clamp subunits become very difficult, and as a result, the DNA polymerase 1 becomes very less efficient. So in prokaryotes, the DNA polymerase 1 do not do the main role of replication. Only the DNA polymerase 3 is doing the main role of replication. In our next video, we'll move on to the molecular details of how the replication happens.